So today we're looking at the required practical for testing food samples, and you need to be able to explain 4 different food tests. Now these food tests you would have done in year 7 and again in year 9 and possibly in year 10. They're not very exciting, however they do produce some pretty colours and they're quite messy, so they're quite good fun. So this is a qualitative test, so you can't quantify the amount of starch or fat or reducing sugars or protein available. All you can do is say yes, they're there, or no, they're not. So that is a qualitative test. So here's the reference for the Caboodle textbook. Knock yourself out. There's a bit more science in there if you're looking for a bit of fun reading. Some nice picture of molecules, some polymers and monomers. Yeah, you know, guys know how to party, so get on with it. So the purpose of the practical. In this practical, you're going to test different foods to see whether or not they contain one of the 4 groups that we need to know about. There's a really useful YouTube video, more useful maybe than my YouTube video, that will tell you how to do this in a bit more detail. As you can see, there's these beautiful colours that I've already talked about across the bottom of the screen. And we're going to talk about which one of those is a positive or a negative test, depending on which food test we're doing. So these are qualitative tests. They give us a yes or no answer. And they're different from quantitative tests that tell you the quantity present. You need to know the name of each test and the correct colour change for the indicator. And we're going to start off with starch, which is using iodine, okay? So simple materials that we're going to start off with. You can have a range of different foods. You probably use cheese, crisps, pasta, ham, maybe some boiled sweets, nuts. Okay. And we're also going to use a spotting tile or a dimple tile, iodine solution, and a pipette. Now the safety here is, and this is quite serious, iodine solution is harmful. So we shouldn't really have contact with your skin. You do need to wear eye protection, and do not eat any of the food because basically it's been in the prep room for years. Okay, some of it even came from Wilderness. It's minging. Don't ever eat the food in science. I know you all think we're saying that because we just don't want to share, and that may be the case, but it's not McDonald's, it's old cornflakes, so don't eat it. So the method, quite a simple method, 5 steps. Again, let's not be writing all this out. Okay, food on tile. That's what I'd put for the first one. Few drops of iodine. Next one, if starch equals black, no starch equals yellow, red. Results in table. Can you see what I'm doing there, guys? Let's not be writing loads. It will help you with the revision. And the results. Basically, the orange iodine solution will turn black blue if in the presence of starch. It is really important that when you start your test that you check that your solution does give you that orange colour, because some Muppets, not that I teach any Muppets in year 10, you know who I'm talking about, occasionally thought that they could drop the food straight into the iodine solution. A, the bottle's brown, you're never going to see a result, and B, it just means that it then colours everything with starch, blue-black, because it's got starch in it. So make sure that you get that orange colour to start with. And the blue-black is a standard phrase that they want to hear when they're talking about this. Right, let's talk about reducing sugars. Okay, so again, we're going to have a awesome range of foods, pasta, ham, etc. And this time we're going to have something called Benedict solution and a water bath, okay, or beakers with supply of hot water, maybe even a kettle, and some sticky labels or waterproof pen. Again, the Benedict solution's got copper sulfate in it, so it's really important that you ensure you wear eye protection, guys. Can't emphasize that enough. And it's a nice bluey solution. So what we see here is that this time we're going to do this in a test tube. We're going to put the food into the test tube, cover the food with Benedict solution, place in a water bath for 10 minutes, record your results. The heat is needed for this reaction to occur. If the water isn't hot, it will not occur, okay? And you might get a variety of changes. So, what we see is when Benedict solution turns brick red on heating, if if a sugar such as glucose is present. The precipitate takes a while to settle in the tube, and you're more likely to see it if it's simply red or brown colour. If there's not much glucose present, you might get a green, a yellow or an orange, but the answer they're really looking for is brick red or red yellow. Okay? All of these colours give you a positive result, but in the exam, when they're going to be assessing this skill, they're going to be looking for the term brick red. Okay? It only tests for glucose, it doesn't test for sucrose. Okay? So it would be no good doing this with certain sugars. And if they say to you, why can't you do it for sucrose? It's because it's a non-reducing sugar. And those of you coming on to see us in year 12 and 13 to do biology, we're going to do lots about that then. And you're really going to enjoy it. It's a really good, fun practical. All right. So, next one, testing for lipids. So, test number 3, it's the emulsion test. And again, we've got our range of foods. We've got a test tube, rack, a test tube, ethanol, water, and a pipette. Here's my method. Try and reduce each one of those into 3 words. Just try and condense that down so we're not trying to remember massive, great big chunks of information. Ethanol is highly flammable. Okay? So therefore, we need to make sure there is no contact with naked flames. If you used a Bunsen to make your water bath in the previous practical, you cannot have the ethanol out with this one at the same time because ethanol vapour is highly flammable. And what we do is we put our food in the bottom, we then put a few drops of ethanol and we shake our test tube and leave it for a minute. And then we pour the solution into a test tube of water. Now what you will see, if lipids are present, the solution goes cloudy with a white layer. Oh, sorry, I didn't mean to go that fast. If there's nothing available, it just goes clear with chunks of food in it. You need to be careful because some food just goes mushy. But we are looking for a nice cloudy precipitate. Alright? So, let's move on to the last exciting test, which is testing for proteins. So, the equipment and small materials that we need to use are our range of foods, test tube racks, this time a biuret reagent, and you can either have this as biuret together or 2 separate reagents, which are sodium hydroxide and copper sulfate. Again, is our trusty pipette. 5-step method. So, what we need to do here is recall our method. Chunk that down into 3 words for each bullet point. Spot the deliberate mistake. There's not 5 points, there's only 4. Anyway, so, again, the biuret reagent is an irritant. The sodium hydroxide, which makes up the biuret, is an irritant. The copper sulfate solution is also an irritant. So therefore, we need to wear eye protection, and any spillages or any contact with skin, it needs to be washed straight away. And as already said, don't eat any of the food because there's the risk of cross-contamination. And I like this one because it gives you a beautiful purple colour if protein is present. Purple for protein. Look at that. A bit of alliteration. Cross-curricular, guys. So, a purple ring between the layers indicates there is protein present. So, what you might see is a chunk of food and then a very fine purple layer between the two. And if that's the case, then there is a small amount of protein, but there is protein there. It could be the fact that there is oil available, and that is also stopping it becoming a solution. So, you do need to examine it quite carefully. Blue biuret reagent turns purple if present, and the purple ring between the layers also indicates that the protein is present. Right, so here's a quick summary of how to improve accuracy. To get accurate food test results, we need to prepare the suspension of food, and to do this you need to follow the following steps. Use a pestle and mortar to grind up the food. Okay, that's the thing that looks like a bowl with a little stick. And we mix 2 spatulas of food with 2 centimetres of distilled water and shake for a minute. and we filter the suspension into a funnel into a clean test tube. Now you're thinking, why is she on about this? But essentially, some of these foods don't break up very easily. If you try and test cheese, it doesn't really mush particularly well. But if you make it into a solution and add the water and give it a really good shake, and then filter out the chunks of cheese, then what you are left with is all the soluble bits of the food inside the water, and you can test whether or not there's any protein, reducing sugar, or starch present. Okay, and you use the filtrate in a clean test tube for the food test. And this is one of the reasons that when you go on to A-level, you do it this way rather than doing it in dimple tiles, because it gives you a more accurate result. You're not just looking at the chunks of food, and those chunks of food that I talked about being suspended in bits of cornflakes inside your test tube aren't clouding your result. So, some exam style questions, because I know that you quite like those. So 3 of them. Can you just pause the slide now and answer these questions for me? And here are the answers. Give yourself a score. Some useful links for you to go on and have a look to make sure that you then can see how this is related to other parts of the science that we're learning, especially the digestive system. There is a spelling test for this activity. Okay guys, and I'll see you tomorrow. Alright then, bye.